protein loop.
An irregular secondary structure in protein that formed from 2 to 16 residues. It is a diverse class of secondary structures comprising turns, random coils, and strands which connect the main secondary structures, alpha helix and beta sheet. Turns are the third most common secondary structure elements in proteins, and up to 1/3 of residues may be involved in turns in a globular protein. As their name suggests, they are responsible for changing the direction of the polypeptide chain within a globular protein. Most turns consist of up to 4 residues. Long loops are also sometimes called random coils, although they are neither random, as they exhibit some amino acid preferences and some structural preferences, nor coiled like a helix. Loop can be found on surface of molecule and contain polar residues, which participates in forming binding sites and enzyme active sites. Protein loops can function in 4 different ways: forming enzyme active site, as recognition site for antigen, ligand binding site, and also protein-DNA interaction. A protein's function depends on the ability of its loops to adopt different conformations. Loop closure is essential for the catalytic mechanism of triacylphosphate isomerase. When the substrate binds, a loop changes from an open conformation showing in pink, to a closed one showing in green, which prevents solvent access to the active site and stabilizes the intermediate compound of the reaction. Loops are important for an antibody's function, since its ability to bind to an antigen is mainly determined by 6 hypervariable loops. The huge diversity in structure displayed by these loops is the key to how antibodies can bind to such different substances. Loops from F-hand are ideal for grasping other molecules. The basic F-hand consists of 12 residue loop region between, forming a single calcium binding site. Calcium ions interact with residues contained within the loop region. The interesting part here is protein-DNA interaction, and we can see from here is the interaction of 3 loops of methyltransferase with DNA clearly. This changes the conformation of the protein, which brings the 3 domains of protein interact with major and minor groove of DNA. There are 5 most well-documented classes of protein loops. These classifications are designated based on the secondary structure these loops attaches to. These classes can be further divided into many subclasses depending on their geometry. The alpha-alpha loop refers to those flanked by 2 alpha helices. The alpha-beta loop stems from alpha helix and connects to beta strand. The beta-alpha loop, on the other hand, is assigned to loop connecting beta strand and alpha helix. The beta-beta link refers to 2 parallel, but non-adjacent beta strands. The beta hairpin is shaped like so, hence the name. This loop is flanked by 2 adjacent antiparallel beta strands. Over time, new classes emerge from the 4th most commonly found secondary structure, the 3,10 helix. The 3,10 helix is referred to as a super-secondary structure and differs from alpha helix in terms of the hydrogen bonds. The regular alpha helix forms hydrogen bond between the I+4 residues, while the 3,10 helix forms the bond between the I+3 residues. Loops between the 3,10 helices, beta strands, alpha strands, and another 3,10 helix are designated similar to the classic loops. There are many other specialized loops documented, and among the more widely discussed are the omega loop. The loop forms are longer with smaller distance between their hinges, that connects to the secondary structure, and formed a shape similar to the Greek letter omega. Protein loop structure prediction is the bottleneck in solving protein structure prediction. Most of the time the comparative protein structure prediction is limited by the errors in alignment and loop modeling. Generally there are 2 approaches involved in protein loop structure prediction. The first approach would be template-based, whereby it uses a database approach that searches for the segment of main chain that forms a loop. The search is performed through a database, for instance FRED, which consists of known protein structure, including the homologs with the modeled protein. The loop predicted are sorted according to the geometric criteria, and the sequence similarity between template and target loop sequence. Meanwhile, the second approach is the non-template based, also known as ab initio. The loop is predicted based on the conformational search or the enumeration of conformation in the given environment. The loop prediction is guided by the scoring and energy function. The loop could be predicted using a web-based tool, modeler,